It's a pleasure for me to introduce Marty Walsh, who has come to us from New York City. He is at Mount Sinai at the Icahn School of Medicine at Mount Sinai. He has many hats there. He is a professor and director of the Center for RNA Biology and Medicine, co-director of what is that fund?
NIH Common Fund.
The NIH, well, I know that, but it was a big abbreviation.
Molecular Transducers of Physical Activity Consortium.
I was supposed to know this before I...
That's okay.
So for the NIH Common Fund analysis site for genomics, he's interested in epigenomics, transcriptomics. He is a professor in multiple departments there. A little bit about his background. He got his PhD from Columbia, and his first postdoc was with Jim Watson. For the young people in the audience, anybody know who Jim Watson is? I didn't hear a yes. And that was at Cold Spring Harbor, and went through his postdoc and ultimately ended up at Mount Sinai, where he is currently a professor, as I said, in pediatrics, pharmacologic sciences, genetics and genomic sciences. And from the title of his talk, I used to think I knew how RNA worked. DNA, RNA, and a protein, and that isn't even close. And so he is going to talk to us today about long non-coding RNAs and how they impact on cell fate. And since you've heard enough from me, why don't we welcome Marty to the talk. Thank you, Marty, for coming. And you have which one?
So can everybody hear me? I'll make sure. So again, I personally want to thank Jim for the invitation, and I certainly want to thank you all for coming on this chilly but beautiful morning in Minnesota. So as Jim mentioned, I'm involved in a couple of different projects and programs, but still near and dear to my heart since my days at Cold Spring Harbor has been working out understanding the basic principles of what directs cell fate, particularly as it pertains to cancer. And so these are stories, and they're ongoing studies. And amid our travels towards looking at the role of non-coding RNAs, enhancer RNAs, there is the emergence of these controversies about what they do. Are they really functional? Are some of these RNAs functional, or are they not? And what's their role in biology, such as it applies to cancer biology and stem cell biology? And recently we linked some of these through a protein called PGC1 alpha, which is also an important modulator of metabolism, as many of you might know. So again, I just want to acknowledge my group. Some of the people here have left, but today's talk is really going to be with Fawn in mind, Side Lee, who just recently retired from my lab after 25 years, Serena Di Cecilia, and others like Ife and Namisha, who are still part of the lab and being quite productive. So again, I need not have to tell you about the dogma that we have here, where DNA is transcribed into RNA, and that is then translated into protein. The question that we asked is, what's the role and purpose of all these pervasive RNAs that are being transcribed as non-coding RNAs? And there's some controversy about whether or not they are capable of encoding short peptides to actually playing a fundamental role in navigating nuclear architecture, and even in the cytoplasm, do they play some type of role as scaffolding and other attributes that remain unclear. So there are many, many resources now that one can go to, and one of those that we've taken advantage of is the Phantom dataset, which is constantly being updated, in addition to those resources through ENCODE and other initiatives that have been found. Again, this is to highlight a paper that came out from Robin Anderson's group about the role of how enhancers ultimately become transcribed, and does that correspond with coordinating transcription at a larger level. Now, I know this is it's supposed to be a grand rounds talk, and so I'll try and throw in some clinical stuff. My lab is also involved in in some clinical studies where we're doing single cell. Unfortunately, I won't have time to talk about some of the data there, but I'll give you a little tidbit about some of the things that we're doing. But this all started from a study that I was engaged with my friend and colleague Mingming Zhu, who is chairman of the Department of Pharmacological Sciences now, where we tried to understand the role of how one non-coding RNA called ANREL, it stands for antisense non-coding RNA to the ink locus. And basically the model here, this was published in 2010, and it was really the first detailed mechanistic understanding where we actually did both genomic analysis and structural basis of how this protein CBX7 was engaged with a non-coding RNA called ANREL that ultimately provoked the repression of P16 through this interaction of ANREL with CBX7 through a very specific secondary structure in RNA. Since then, we've been testing the relationship not only of that, but how RNAs are modified. And in a paper we published a few years ago in Cell Stem Cell, we found that there's a direct relationship between an oncogenic transcription factor called ZNF217 and this enzyme that acts as a methyltransferase for adenine called METTL3 or METTL3, which also is a heterodimer with METTL14. So the idea was that METTL3 and ZNF217 interact to abrogate the methyltransferase activity of this enzyme to maintain the stability of RNAs. In another study we published not so long ago, also with the help of Zisca Aguilo and Side Lee, we showed that in fact PGC1 alpha, a transcriptional coactivator, can also evoke methylation through methyl-5 cytosine in some enhancer RNAs. And this directly correlated with both enhancer RNA stability, but also seemed to regulate a core set of metabolic genes such as sirtuin-5 and some other enzymes like IDH3A. And then today what I'm going to mainly talk about is our recent work to be published, I presume, very shortly in Nature Genetics about this enzyme, about this protein WDR74 and its interaction and link with EZH2 beta-catenin and its potential role in orchestrating the transcription of MYC over very long distances. So again, this is a cartoon derived from Jeff Rosenfeld's group from a review in Nature Review Genetics, and basically they tend to argue the point that enhancers are ultimately transcribed vis-a-vis through RNA polymerase II. They, like many RNAs, they undergo capping, and then they are ultimately utilized by various RNA binding proteins, and most recently by the Shekatar laboratory showed that this complex between these RNAs and integrator enhances the transcriptional output by RNA polymerase III. Where we came in, we showed that in fact PGC1 alpha and this protein of the ensign family called ensign 7 as a methyltransferase could methylate 5-methylcytosine, thereby stabilizing these enhancer RNAs. More recently, we're interested in understanding this relationship of WDR77 to this cat binding complex, and this, in fact, may also perpetuate the capping of specific enhancer RNAs as well as other transcripts. So again, enhancers, as you know, become quite the, I guess, the new trend now of looking at all of these SNPs and questioning whether or not they are functioning as enhancers. Are they promoting gain of function, loss of function of various circumstances? What we now know is that enhancers communicate with all aspects of gene transcription. That includes initiation or pre-initiation, transcriptional initiation, then there's also been much to do about how enhancers communicate vis-a-vis through PTEF-B and BRD4 to cause a thing to process pause releasing of the transcriptional apparatus. And then there's been more recent studies to show that, in fact, by the Shelley-Fatard group and others, to show that enhancers also communicate with the elongation process as well as involving co-transcriptional splicing. And then finally, Nick Proudfoot and others have shown that enhancers also play a role in how genes are ultimately transcribed and how those transcriptional modules are terminated through various communication aspects with the enhancer. So enhancers play fundamental roles in all aspects of transcriptional control.
Marty, is this all the same enhancer that does all this? Or
Well, this is just general logic, but not all enhancers do this. The role is, again, as I mentioned, enhancers can come in all sorts of flavors, but again, enhancers could also include super enhancers, which really are extensions of single enhancers being multiplexed over long distances using. So we took on a model in collaboration with folks in Toronto, Hansen, Hay, and others, where we tried to understand the relationship of long non-coding RNAs as it pertains in prostate cancer. And this is just a cartoon from a fairly recent review, 2013, where you have a benign prostate. This can actually transform into high-grade, basically they call high-grade PIN or prostatic intraepithelial neoplasia. During this time point, we start to see a change in the rate of proliferation of certain epithelial cells. This results in the increase of these non-coding RNAs, as well as the loss of these microRNAs, miR-23B and others. But what you start to see is basically the evolution and the elaboration of how long non-coding RNAs ultimately get dysregulated in their contribution to directing cell fate. And so our work really of late was looking at the role of PCAT1 and this other gene called PCAT8 or PRNCR1. And this ultimately leads, their expression and abundance corresponds with localized prostate cancer basically transforming into metastatic prostate cancer. And so we think they play a fundamental role. What's really noteworthy about these two genes is that they sit on the axis of MYC, and in a paper that we published jointly with Hansen Hay, we showed that in fact one risk allele called RS74637008 had a conversion, and this conversion resulted in the gain of function for recruitment of 1CUT2 that then made this gene much more aggressive in AR responsiveness. PCAT itself as a long non-coding RNA was then shown to interact with AR and LSD1 to promote these androgen-responsive genes, GMNT1 and others, that ultimately led to transformation of prostate epithelial cells. So again, I'm not going to go over that. In our study, another study that we performed, a long non-coding RNA called RBM5AS1, we showed that in fact it was linked to stabilizing beta-catenin on TCF4 sites that activated genes such as MYC, YAP1, and SGK1. So I'm not going to spend any time on that, but I'm going to basically go over some of the things that we've been interested of late. So basically at the time we started this study, we decided to use an approach called laser capture microdissection to capture these basically ducts and transform ducts, and then we proceeded with doing sequencing, and then this led to us being able to analyze the genes that were corresponding with changes in their invasive capacities. And so what we found when we compared invasive prostate cancer compared to the surrounding normal ducts, we saw that among the genes most highly upregulated was this PCAT1 and PCAT8. This is an old story now because these have been long since known by Sinai and other groups that have shown PCAT1 and 8 being very important as a registration for the invasiveness of prostate cancer. But one thing we did notice is that there was a relationship between these long non-coding RNAs and their neighborhood and the genes that surround them. And one of the things that we took from this information is that we saw the majority of long non-coding RNAs, the genes that surrounded them also correspondingly went up, or if the long non-coding RNA was down, so was its neighborhood gene. However, there are a couple of circumstances where that logic was defied, one being P10, the long non-coding RNA right next to it was slightly upregulated, and this corresponds with its downregulation. In our case, I mentioned ANRIL being an example of that. We see ANRIL here and its neighboring gene or overlapping gene CDKN2A or INK4A being downregulated. And there are other circumstances where other long non-coding RNAs being down, like NKX 3.1. So this started to build some type of logic that there was probably a relationship between neighborhood genes. And the question is for these non-coding genes, the existing promoter, you see that they may influence genes that are surrounding their neighborhood, in their neighborhood, such as this promoter and this promoter being actuated through this transcriptional unit. And so the argument could be is maybe what's happening with some of these non-coding genes or long non-coding genes is that their promoters may in fact be switching over and functioning as enhancers for these neighborhood genes. And this could also take root in the fact that some of these enhancers may also have a negative influence on transcription of certain genes as well. But we think this is a much less likely case. So again, this is just to rehash what we did. Basically, you see in 2 patients that we had access to their tissue from. We took normal surrounding tissue ducts and compared that with those that were involved with invasive carcinoma. And you can see that these genes were dramatically up or elevated. Again, this is not really a surprise, but sort of reinforce the notion of that these so-called neighborhoods were likely impacted. Again, this is just looking from TCGA data sets that we showed that, in fact, statistically significant is the abundance of PCAT1 when we compared 38 sets between normal and invasive carcinoma, and we can see that there's a statistically significant elevation in almost all the samples that we looked at that had normal, and this is also true of PCAT8 shown here. So again, this made a case that their elevation was corresponding with the incidence of invasive carcinoma. Again, what was also a surprise to us, and you know, after looking at the data quite carefully, this is not our data. This was published by Arul Shanayan's lab at the University of Michigan. Basically, what was known as a super enhancer shown here. Again, you can see the enrichment of this transcriptional coactivator that typically marks enhancers. You see that it's elevated over the super enhancer. When we add JQ1, it is diminished, and this also marks H3K27 acetylation in the same sample shown here. You can see this enrichment overlapping MYC and another gene that is quite known here called PVT1. But what was really fascinating to us was that we saw this remarkable enrichment of BRD4, remarkable enrichment of H3K27 acetylation that was sitting over these genes that were noted for being quite abundant in prostatic carcinoma. And these were all performed in these VCAP cells, which are like LNCAP cells but are androgen unresponsive. So the argument is this distance sits roughly about 750 kilobases from the MYC oncogene, which is noted to be in what is regarded as a gene desert. MYC is been one of the most difficult challenges to drug, and so the question is what aspects of MYC have been utilized to drug, such as BRD4 inhibitors and others. But the question is this relationship makes it quite interesting that in this gene desert, there's many non-coding RNAs that are transcribed. Of recent was the CCAT2, which is called colon carcinoma-associated transcript 2, has been shown to directly link with the MYC promoter to drive MYC abundance in colon cancer, as well CCAT1 as well has also been linked to MYC activation, and recently others have shown that this is a plausible consideration. So what we set out to do was that we engineered an aptamer that sits at the 3' end of PCAT1. This was synthesizing an aptamer that could bind streptomycin. And so we used this as a means to be able to capture these long non-coding RNAs. We did this for PCAT1, PCAT8, and as well as PBT1. And the idea was to engineer this by CRISPR these tags, leading to, in this case, one for PCAT1, that we engineered this streptomycin binding tag or aptamer. And we actually were able to use it to capture proteins that were selectively bound to this long non-coding RNA. And what was quite remarkable from this was that we could reproducibly see, and this was actually shared not only by PCAT1, but as well as PCAT8, and to a lesser degree, but also equivalent was PBT1, that we saw sort of this elaboration of a mixture of RNA binding proteins, but also many of these RNA binding proteins were also intersecting with transcriptional activators. Some of these proteins were as well linked to transcription elongation, such as the PAF1 complex, AF9, AF4, which made this quite interesting. And another one that was kind of an oddball, but we didn't know what it was about, was this WDR74. And there's really not much literature at the time about this, but the other thing that also came out reproducibly were components of the capping complex, NCBP1 and NCBP2. And so in order to confirm that, we used our streptomycin pull down of TCAT1, and then we immunoblotted for WDR74. This is the input compared to the RNA bound. You can see enrichment of the protein being pulled down by PCAT1. The same is true with another cap binding protein, NCBP1. We see that we could pull down this protein selectively. And what was also interesting, EZH2. Now I should note that despite the paucity of information about WDR74, it was noted to be one of the closest homologs to another WD40 protein called EED, which is a component of the PRC2 complex, which also could bind EZH2. So the argument was that possibly WDR74 might be replacing EED in stabilizing EZH2, because in the absence of SUS12 and EED ultimately leads to the degradation of EZH2 in other systems. So this has been postulated by Miles Brown at Dana-Farber and others, but we tried to give some inclination of that this might be a mechanism that salvages EZH2 that is highly abundant in prostate cancer. So another noteworthy thing about WDR74, it was among one of the promoters most heavily mutated in cancer. From a paper came from Bill Lee's lab. They looked at genome-wide analysis of non-coding regulatory mutations in cancer, and of those non-coding mutations, in addition to TERT and PLEC-H1, HS1, WDR74 was among the most highly mutated promoters. And of those mutations, we looked at them very carefully from the data that they provided, and you could see this is sort of where they clustered mainly around the promoter of WDR74. What was really quite remarkable about all of those mutations was they ultimately led to basically a gain-of-function mutation, leading to developing a motif of the ETS family. And so this provided some evidence that maybe what's happening is that WDR77 that's mutated, many of these mutations are gain-of-function mutations that drive expression of WDR74 in a wide array of different cancer types. And to our effect, we did both grow-seq and chip-seq, and what we found was that of the strongest eRNA signals that we saw, WDR74 was the target or the gene most closely linked to this eRNA. This is just from our chip-seq data of EZH2 and EZH1, as well as some, this is actually RNA-seq data. I don't have the grow-seq data here, but the grow-seq data also showed a very strong signal that was associated with this gene WDR74. So what is it about WDR74? We did some Kaplan-Meier curves. Again, we don't have prostate cancer here because many of the patients with prostate cancer survived, but we did perform a correlation in triple-negative breast cancer, renal clear cell carcinoma, and glioblastoma multiforme. What we found was that the overabundance of WDR74 corresponded very strongly with poor survivorship. And this was noted in other cancers as well as non-small cell lung carcinoma and others. But again, this highlights the fact that WDR74 expression corresponds with poor outcomes and survival. And again, just going back to what we saw in the interactome, WDR74 had interacted with PCAT1 interacted with a large array of different proteins, mainly of which included many RNA binding proteins. But what was also interesting was that WDR74, when we did a direct pull down after we made an antibody, we were able to show that in fact it interacted very strongly with beta-catenin. It interacted somewhat with TCF4, but this blot was not really the best blot that we did at the time. EZH2, we saw a very strong interaction. So again, this reminded us of our own study that beta-catenin itself may also play a role in being recruited by PCAT1, as we showed with its role with another long non-coding RNA, RBM5 AS1. And again, we showed that WDR74 interacts with beta-catenin through pull downs that we did as well. It's highly expressed in LNCap, where we took a non-cancerous cell line RWPE1. You see it's expressed, but barely abundant as it is in the LNCap cell line. So again, this building out the interactome hierarchy here, we see that WDR74 is at the apex of its interaction with PCAT1. So the question is what is it actually doing? I noted in our mass spec data that much of what was coming down also from the PCAT1 pull down was that we saw many cap methyl 7-guanosine cap binding proteins that were associated with this interactome. So the question that we ask is, what happens if we knock down WDR74? What ends up happening in the neighborhood, as I pointed out? And by doing so, we used hairpins at the time, and so we used two types of hairpins. One was a translational interrupter, and another one, or translational disruptor, and another one was directing cleavage of the protein of the RNA itself. What happens is that by knocking it down, you can see a depletion of the protein in both circumstances. As well, we also did a knockdown of EZH1 using the same translational inhibitor as well as a dicer-mediated inhibition, and we can see the loss here. What ends up happening is that when you knock these things down, the long non-coding RNAs go down. PCAT1, PCAT8, PVT1. But what was really interesting, and I apologize for not having the data here, but the growth-seq signals or the nascent RNA signals were not at all deterred. So it sort of indicated that what might be happening is that there might be some issues with RNA stability, and that pointed us in the direction that maybe what was happening was that capping might be altered. As well, I should point out that these other long non-coding RNAs as well, which are also highly expressed in the LNCap cell line, were also diminished by knocking down EZH2 and WDR74. So this pointed a picture that maybe what's going on is WDR74 may be influencing capping, maybe splicing. It's altering at some level, the steady state levels of these non-coding RNA transcripts. And so shown here, again, just to make note, is that EZH2 in the same 37 paired samples, we saw EZH2 is very much significantly elevated, as is MYC, and as well as WDR74, albeit less so, at DRD4 as well. So this argued somewhat that obviously WDR74 is changing the long non-coding RNA landscape surrounding the MYC oncogene. And so we go back to some data that was published. This is really derived from Miles Brown's lab, where they tried to argue that, in fact, EZH2 may have a non-canonical activity independent of PRC2 that may evoke transcription over MYC. And this has been contested by a lot of people, but I think there's some relevance of that postulate in the sense that EZH2 has to go somewhere, and we believe that it gets stabilized at some level. Its abundance is there, and it may not necessarily have to associate with PRC2 to evoke this type of response. And so what we did was that, again, we knocked down EZH2, and basically, no surprise, we lose the chip signal. But what was also interesting is when we knock down EZH2, we also lose this WDR74 signal across different regions surrounding the MYC oncogene. Postulating again that what might also be going on is that WDR74, like EZH2, are sort of tethered together and might be interdependent on ensuring the stability of how these long non-coding RNAs are presented and stabilized across this landscape. So again, this distance is quite long. It's roughly about 600 kilobases. How does one analyze relationships like this? We took the approach using gapmers to try and disrupt or basically remove these long non-coding RNAs. They're a little challenging to work with. We worked with this company called Icahn, and then more recently we worked with Ionis Pharma in designing what are called integramers or whatever, gapmers, in being able to remove these RNAs from this cell line. This is an LNCap cell line that's actually made to be castrate independent or castrate resistant. Thank you. Resistant cell line. We tested in that cell line. What we could see is that we can readily remove PCAT1 with up to 500 picomoles of gapmer. 200, we see a reduction. But what this resulted in is a dramatic loss in the PCAT1 expression, but it also corresponded with a loss in c-MYC as well as in PCAT8. So when you knock down these two lincRNAs using these gapmers, you can dramatically reduce MYC activity. Now again, this is not a surprise because Arul Shanayan's lab had shown previously that these two lincRNAs were associated with MYC abundance, but again, this sort of makes the case that there's something in addition to what they were postulating that might be going on. So we used an approach, as I mentioned, we're interested in looking at these long intersections between the genes that were up here to the genes down here, such as MYC, and we used an approach called Hi-C. Basically, it's a system that can capture looping events between enhancers and promoters over long distances. It's really not designed to capture short intersections or interactions of distances less than 100 kilobases. However, any distances longer than 100 kilobases are easily captured using this method. So basically, you do restriction digest. The ends of the restriction digest are biotinylated. They undergo ligation. You reverse the cross-links, and then you engineer a Hi-C library, and then you sequence, and then what you do is you capture what are called topologically associated domains, highlighted by these so-called triangles that you see emerging here that sort of define proximity of these ends to one another to be able to see if you can map out specific interactions in these so-called topological domains. And that's precisely what we did. And so we're able to show that, in fact, using high C, we're able to capture what is called a TAD, or a topologically associated domain, between PCAT1 and MYC. This is just showing just the relative collision frequency that occurs. We did multiple repeats, and that resulted in this type of map. What we then proceeded to do is to use somewhat of a fish study that did a contrast where we labeled the PCAT1 RNA with MYC DNA. You can see in the RWPE, you can see that they're closely linked together, but they're not necessarily compacted or consolidated. However, when we compared that with the LNCAP C42 cells, you can see that this is much more consolidated, and that we use a measurement called collision frequency index. And you can see that this is a much higher ratio, and so we can see that, in fact, when we measure this compared between the two cell lines, and you basically ablate that when you basically knock out the RNA in this cell line. So again, the concept is that these lincRNAs may actually be driving these looping events. And there's been some evidence in other models that have stressed this type of relationship. So again, we just compared that using chromosomal conformation capture, or 3C. This is just a more direct measurement of looking at these looping events between c-MYC, PCAT1, and PCAT8. And again, just to highlight a couple of things, that we think each of these long non-coding RNAs are independent in forming distinct loops. For example, a PCAT8 loop, or PRNCR1 loop with MYC, is dependent on that lincRNA and not PCAT1. So when you knock down this lincRNA, it deters or diminishes the looping frequency. When you knock down PCAT1, it diminishes this looping frequency. So again, these loops seem to be very specific from the RNAs for which they're being transcribed from. So it may be that the transcription of these things are interdependent or dependent with c-MYC transcription, and that the resulting RNA might play a role in extending these loops. Again, when we knock down the WDR74, we also diminish the frequency of these loops between c-MYC and PCAT1, and as well as PCAT8. So again, these argue the point that the lincRNAs are extending over these enhancers, these so-called enhancer loops, towards the MYC oncogene. And again, just to make a case for their oncogenicity, we basically used the same cell lines. We inoculated them as xenografts, and when we knocked down WDR7, when we compare it to the locked nucleic acid control and compared to a control hairpin, we see that there's diminished growth by the tumors. They still grow and ultimately lead to animals actually having to be sacrificed, because the tumors ultimately do get large enough that we had to sacrifice. But again, this is just losing bioluminescence tracing of the tumor isolates that we recovered, and you can see that the tumors are dramatically different in size when you knock down PCAT1, as well as when we knock down WDR74. And you can see that there's a significant loss in MYC abundance. So with that, we wanted to understand the basis of this. And so we engineered a transcript that was intact and another transcript that was lacking the first 45 nucleotides of the RNA. What ends up happening is that we did a super shift. Basically, it's using an antibody against these two proteins, WDR74, EZH2, and when bound to the RNA, we could measure their selectivity for binding this specific PCAT1 RNA. When we knock out or lose the first 45 nucleotides, we lose the selective binding. So we then tested the idea that maybe what's going on, as I mentioned earlier, is that maybe WDR74 and EZH2 may be also playing a role in capping events. So we performed what are called nuclear run-ons. Basically, this is just to capture nascent RNA. And what we then did was immunoblot for methyl 7-guanosine caps. This was plotted on a mixed cDNA, and you can see here, and I apologize, this is sort of grainy in this picture, but we can see that in the control we see quite abundant capping. However, when we knock down WDR74, we lose significant signal at the caps based on the IP blot. We also see a significant loss in capping by methyl-7-guanosine when we knock down PCAT1, leading to the indication that these lincRNAs and this protein are playing some type of role in capping events. To make this a more clear understanding, we did what is called PAR clip sequencing. WDR74 was used as the bait to capture transcripts that were bound by WDR74. And what we were able to find was that what was quite striking about it was that when we knocked down this PCAT1, we lost the signal for WDR74 to recognize the 5' end of transcripts of the MYC gene as well as the PVT1 gene. So this then indicated that what was likely happening is that WDR74 is recognizing potentially the caps of MYC and PVT1 from this genome-wide interrogation. We then looked at the metadata, and we found that the enrichment of transcripts and where they were bound by WDR74 almost exclusively occurred at the very 5' end of all of the annotated transcripts that we could capture. And this corresponded nicely with NCBP2, which is a known cap maintenance complex binding protein. And you see this little blip, and this blip is typically because the 3' end of transcripts typically loop with the 5' ended genes. And this was sort of the motif analysis showing that this guanosine is likely the methylguanosine cap. So again, just to make this proof of principle further, we then did a knockdown of an enzyme responsible for the methylguanosine cap called RNGTT. When we knock it down, we lose methyl-7-guanosine abundance. This as well, we see virtually complete loss of these caps when we knock down this cap maintenance protein. And interestingly enough, when we knock down WDR74, we see a significant loss in the abundance of this cap across all transcripts. And this is just showing a non-related control RBM5, as I mentioned before. So the question is the active transcription resulting long non-coding RNAs in phase-separated regions? I'm sure you've heard a lot of this in literature, that the nuclei, and this is nothing new, Joel Gall has long postulated in Cajal bodies and other bodies that these various domains in the nuclei are undergoing disordered associations through proteins that are also themselves disordered, leading to these punctated regions in the nucleus. The idea is that maybe some structures, such as paraspeckles that are linked to splicing, we believe that other processes, as I mentioned before, are inclusive of these phase-separated regions, such as capping, that include super enhancers. And so that's been our argument. And so to make that sort of an interesting point, we then looked at using antibodies that we generated against WDR74, and he compared that, and this is generously provided by David Spector at Cold Spring Harbor. We could see SF1 as well as WDR74 share very similar architecture in the nucleus. Again, these are two different stainings, but again, this makes the point that they are in fact potentially phase-separated punctate in the nucleus. And so we argued that, like splicing, like transcription itself, capping is also immersed in these punctated regions. And so we come away with this model that, in fact, what might be happening is that WDR74 might be subverting EZH2 activity, and this also includes the recruitment of beta-catenin through these cap maintenance binding proteins, NCBP1 and 2, that may help to stabilize MYC transcripts. As people know, MYC is highly abundant and rapidly transcribed. However, one of the things that we know about MYC is that their transcripts are incredibly unstable, and the idea is that any level of stability can increase the abundance of MYC translation and MYC protein abundance. So the idea is that this might be another opportunity to target MYC selectively through disrupting capping mechanism. So, and with the last couple of minutes, I'm going to talk about a novel technology we're pursuing. This is basically bacterium or Paramecium basaria. This was done in collaboration with my colleague Ming Zhu. This gets infected by chlorella and it's quite an interesting virus. It's one of the largest viruses known. It's actually a virus, while this is performed by scanning EM, it's also visualized by phase contrast microscopy. So it's such a large virus that you can actually visually see it budding on the Paramecium itself. Now, what makes this virus quite interesting is that it actually shares many eukaryotic domain structured proteins, one of them being the set domain. It carries a protein called VSET. And what's unique about this protein in the chlorella virus is that it's selective in its capacity to methylate H3K27 marks. And the belief is that this virus sort of abrogates transcription, post-transcription, by marking this lysine on histone H3K27, leading to the silencing of chromatin, possibly in the host. And what makes this unique about it is that it functions independent, and it acts as basically as a dimer to methylate these things. So what we engineered was a CRISPR interference model. You're probably familiar with this crab domain construct tethered to the dead Cas9, and this in turn can shut down transcription by recruiting the co-repressor and chromatin modifying complex to shut down transcription of these genes. We engineered a VSET as a dimer, and this in turn then can recruit VSET through the guide RNA to target these H3K27 marks in a selective manner. We've been able to do this in a high throughput screen using this model. So what we were trying to do is we're trying to identify genes that were synthetic lethal for drug resistance. We engineered or generated a cell line which is a triple negative, well, that's debatable too with this cell line, but a triple negative breast cancer cell line. We basically made it a resistant cell line to ladaponib and doxorubicin, as well as to JQ1. We were then able to grow these cells up. We then tiled guides over most long non-coding RNA promoters, and we then tried to identify their growth phenotypes based on this potential, the result of which we were able to screen. And what was remarkable was that we were able to identify a relatively few targets. Now again, we just targeted, using CRISPR screen, we targeted 9,000, roughly about 9,500 lincRNAs. And of that, we found one that was quite linked to chemotherapeutic resistance, because we were able to show that this resulted in a loss of the growth phenotype. Actually, this has become quite our positive control for a lot of things of late, because PPT1 showed up. This is not a surprise, as well as this other lincRNA promoter as a target. And so what we performed was that we showed that this lincRNA might be functioning as an enhancer for these genes here. So what we're able to show in the controls, the non-targeted controls, we don't see any enrichment of K27 trimethylation. However, with these three guide RNAs, we're able to drive trimethylation of K27 over the lincRNA. And what ended up happening was that when we performed RNA-seq, we see that, in fact, these genes are all repressed, corresponding with this lincRNA, indicating that we can engineer this to shut down these 3 genes just merely by enriching for this methyl mark on H3K27. And so this, again, made us test the role of what's going on in response to this CRISPR-I. So we engineered this cell line again using the same cells. I apologize, the group that was supposed to be the negative control, the cages got flooded, and so we lost those mice. But nonetheless, we were able to show that in the absence of DOX, we were able to basically prevent the tumors from growing. This is just using a sgRNA control. The tumors still grow, and this is still under the presence of doxorubicin. And in the doxorubicin negative control, you can see that the tumors grow quite rapidly, independent. And this is just, again, bioluminescence tracing. So I think I'm going to end there because of the time, but just to summarize some of the stuff that we're doing with PGC1 alpha, as I shown here, published in the Cell Reports paper a couple of years ago, we showed that PGC1 alpha is connected to these enzyme family of, they're basically methylcytosine transferases. They can mark these cytosines with methyl 5 cytosine. PGC1 alpha, as you know, is a transcriptional coactivator. It interacts strongly with mediator 17, MED1, as well as the Saga complex to evoke transcription of a wide array of metabolic genes. We became very interested in these genes because they were so enriched with this enhancer RNA. Basically, just the takeaway from that paper was that we're able to show that, in fact, Cert5 has an eRNA that corresponded with the enrichment of PGC1 alpha, had this motif right adjacent to the transcriptional start site of the motifs that were also enriched by PGC1 alpha and this eRNA. We see this LXR, RXR, NRF2, and of course the PPARs generally. So the argument is what does methyl 5-cytosine do in RNA? Again, we became interested in knocking down these eRNAs. When we knock down this eRNA using locked nucleic acids, this ultimately leads to the loss of Cert5. This is just a proof of principle that, in fact, there is an eRNA there using typical outdated northern blotting procedures. So we're able to show that, in fact, you can actually detect this eRNA. And shown here is that this ultimately leads to the slight loss or abundance of Cert5 protein itself. We showed that the Cert5 eRNA is only selective for the Cert5 promoter. So in other words, you can put a different eRNA there upstream or in a trans-regulating fashion. You can show that that eRNA cannot evoke activation of the Cert5 promoter. It's only the Cert5 eRNA that can evoke activation of the Cert5 promoter, and that's shown here. And this is just showing that we can actually induce the eRNA itself using a doxycycline responsive system. So again, just making the point, Cert5 was of interest to us because Cert5 is an important lysine deglutarylase. It's responsible for activating carboxyl phosphate synthase. It's been recently shown that carbamate abundance is potentially an oncometabolite, favors transition to mesenchymal fates. When we actually fast these animals, you see an increase in NSUN1. These are from livers of mice. You can see that NSUN1 abundance goes up slightly in these animals, using 8 animals, and this is consistent with the Cert1 abundance model, and this also corresponds with the total abundance of methyl 5-cytosine in RNA. And so we performed R-looping studies. Basically, this is capturing RNA that's potentially being transcribed as a heteroduplex with the DNA. Basically, you can generate a sequencing library. What we found was that when we knocked down the ensign, we did both ensign 2 and ensign 7, we could see that there was a significant loss in the population of this group of transcripts overlapping. This is just basically metadata, ref-seq genes associated with PGC1 alpha. We see that there's a distinct loss in this drip RNA-seq signal, which means that these are heteroduplex RNAs. They're basically gone, indicating that these enhancer eRNAs are potentially lost. And this is just looking at the over-enrichment. So the question we wanted to find out is if we can capture potential readers. As you know, PGC1 alpha, as well as other things, metabolism in general is sort of at the crux of the epigenome. We know that acetyl-CoA drives histone acetyltransferases and BRDs. Sirtuins require NAD. LSD1 requires FAD, and Jumanji TET protein. These demethylases require alpha-ketoglutarate. So this is just to make the argument that the metabolism is quite closely connected to the epigenome. We found that PGC1 alpha activates SIRT1, SIRT5. PGC1 alpha activates SIRT5. SIRT5 activates fumarase, which converts fumarate into malate. The accumulation of fumarate is known to lead to oncogenesis, and so it's regarded as an oncometabolite. So we wanted to see if we can capture readers of each of these methyl marks in cytosine. And so the idea was to do a screen. This was performed in conjunction with Brian Strahl at UNC. And what we were able to find was that we could identify selective readers for 5-methylcytosine. We haven't done yet hydroxymethylcytosine, but this one protein is a tandem tutor domain protein called TDRKH, as well as those of the C0 family, H02, as a transcriptional coactivator, and also adenylase complex protein. So these are just some of the questions we were asking at the time, and so we think that 5-methylcytosine, as well as hydroxy 5-methylcytosine, and readers can probably play a role in regulating enhancers. We think that there's a sort of a intersection between enhancers, metabolism, and how these modifications in RNAs are modified. So I think I want to end there. I just want to acknowledge folks in my lab. Siska Aguilo, she has her own lab now in Sweden, in Umeå. Fan Zhang has her own lab in Harbin. Almu just joined my lab about a year ago. Ife was a former PhD student, now a postdoc in the lab. And I just want to acknowledge my very close collaborators as friends and colleagues. And of course, I want to acknowledge some funding that NIH has so generously provided. And again, I also want to make a pitch for our Center for RNA Biology. We're basically interested for computational bioinformatics, postdoctoral and staff scientific positions. So with that, thank you all for...
Thank you. We have time for maybe one question. We have a meeting at 9, I know,
Yeah.
so we can go a little over.
Yeah.
Okay.
So on the PCAT1 binding proteins, it's very promiscuous, whatever the right word is, and is there preferential binding to any one of those proteins? It's sort of hard to figure out because this is happening with other like molecules, and so you've got 100-dimensional orchestra playing probably, and it's, at least for me, it's hard to figure out where the control gets in.
Well, the question that you're asking is selectivity among the proteins for their target RNAs.
Right.
I agree. I think that's part of the, I guess you could say that's part of the controversy in the long non-coding RNA field, and their abilities to evoke different cellular processes. So again, what I showed in some of the earlier things that we were looking at, these were basically several trials that we performed using tagged RNAs.
Yeah.
Again, this is replicates of like 5 IPs using, and again, this is shared proteins between, well, of multiple experiments, but we also have tiled with that PCAT8 and PVT1, and they all share some of the same protein binding characteristics, which makes it kind of interesting because we think it has more to do with the neighborhood, as I mentioned, rather than independent RNAs themselves. So we think that the aggregation of these proteins are sort of confined to what I regard as these phase-separated regions, sort of like enhancer proteins. They sort of like to aggregate together. So that's been sort of the perception that others, like Rick Young and others, have had in the field in how these are organized as domains in the nucleus. And we think capping is one of those structures. David?
So I guess I was confused about the role of EZH2 in the capping complex that you isolated and characterized. So is it not, in that setting, is it not methylating histones nearby? And it's not. And is its enzymatic activity required at all?
No,
So,
it's independent of PRC2 function. And this has been argued by a lot in the field, but Miles Brown has thrown out this postulate. Some people have tested it, has been able to reproduce some of this information, others have not. So it's still a contentious argument over the role of EZH2. You know, my good friend and colleague Danny Reinberg is constantly berating me over the fact that you can't have EZH2 without EED and SUS12. But others have shown that it's there. It's there independent of these proteins. So the question is what's stabilizing? Even a little bit might play a significant role. We know EZH2 is overexpressed in a wide array of tumors, particularly prostate cancer. So the question is what's happening to what's remaining that's stabilized in the nucleus, and if it is playing a non-canonical activity independent of PRC2. And that's sort of what we're arguing, that we think EZH2 might be stabilizing. And it also argues to the fidelity at which one might want to use EZH2 inhibitors. So these
I
are...
was just wondering if you looked at any of these capping activities in the presence of the EZH2 inhibitor or in
We are doing...
activated.
Yeah,
So
we are...
EZH2 has activating mutations in some
Correct.
lymphomas, so
Right.
I'd be curious to know, because maybe it's got another substrate.
That's very possible. Quite honestly, we are testing that. So we have Jian Jin in our lab from UN, who recently came, who designed some of the first EZH inhibitors, and so we're now testing that, as you said.
Okay, that's it. We're out of time, but thank you so much.